Thanks for having me. So I'm going to tell you a little targeted Iso-Seq story from my graduate research at Johns Hopkins, studying this telomere-binding protein TIN2. So just a review on telomeres for y'all who don't think about it. Telomere link's really important for cells that need to divide many times. And telomeres in humans consist of about 8 kilobases of repeated TTA-GGG sequence. That's double-stranded, and then there's a single-stranded overhang of more TTA-GGG sequence. And the whole length of the telomere sequence is bound by this collection of proteins collectively termed Shelterin. And that Shelterin is really important for maintaining telomere length and protecting the chromosome ends. So every time a cell divides, you lose a little bit of sequence off the end of the chromosome. And that's generally okay because telomeres are repetitive and they're non-coding. But over time, if you lose too much of this telomere sequence, they lose their protective ability and then become recognized as DNA damage, which can result in cellular senescence or cell death. And cells that need to divide a lot express an enzyme called telomerase that can add back telomere sequence to the chromosome ends and restore telomere length to somewhere around where it needs to be. And this is a constant balance between telomere loss and telomere addition, resulting in a heterogeneous population of telomere lengths in individual cells and in a population. So the human population is a great example of telomere length heterogeneity. So this is a diagram generated by sampling telomere length in a large number of healthy individuals. And so by doing this, we were able to determine the limits of normal telomere length. So these are percentiles on the graph. So if you sampled most of us in this room, right, we'd probably fall along the green lines, like those black dots. And on the higher end of telomere length, some rare genetic mutations can cause increased telomere length. You see that in cancer, familial cancers. And then on the lower end of telomere length, you get short telomere syndromes. And this is a set of degenerative diseases caused by inability to maintain normal tissue homeostasis. And most short telomere syndromes are caused by mutations in the telomerase enzyme itself or other genes that help regulate telomerase expression and localization and function at telomeres. But we wanted to study a specific telomere protein called TIN2 that's mutated in short telomere syndromes that never had, it's part of the Shelterin complex and never really had any link to telomerase activity directly. So we wanted to figure out why TIN2 would cause short telomere syndromes. So this is a diagram again of the Shelterin complex, and TIN2 is generally considered as a bridging molecule. So it binds the TRF1, TRF2 proteins that bind the double-stranded telomeric sequence and the TP1-POT1 complex that binds the single-stranded telomere sequence, so sort of a bridging molecule, right? And TP1-POT1 is also a telomerase processivity factor, so we thought that that was a good candidate for studying how TIN2 regulates telomere length. But before I get into that, I want to talk a little bit more about the TIN2 protein itself. So this is a schematic of the TIN2 protein, and below it is conservation across the amino acids. And I like to think of TIN2 as being divided into 2 regions. So the N-terminus has all of the Shelterin interacting regions, so where it binds TRF1, TRF2, and TPP1. And just outside of the Shelterin interacting region, we have the cluster where all of the patient mutations fall. And so that's in a conserved domain of unknown function. We have no idea what it's doing. And in addition to that, in the C-terminal region of TIN2, there's this C-terminal extension that's found in one isoform, but not in the other isoform, which I meant to mention earlier, but there are 2 isoforms of TIN2 that are described. And in the C-terminal extension, there's actually a really highly conserved patch of amino acids that we don't know what it's doing. So to look a little more closely at the TIN2 gene, it's kind of interesting. It's really small. It's only 3 kilobases. It has 9 exons, and the introns range from 80 to 400 base pairs. That's about it. And they're shown to scale in this diagram. So the short isoform of TIN2 retains the last 3 introns, and it results in a stop codon right after exon 6. And the longer isoform splices together all 9 of the exons, and then you get the longer protein. And exons 1 through 6 encode the Shelterin interacting region and the patient mutations. So we wanted to study these a little better, but there wasn't really any good commercially available TIN2 antibody to look at protein levels in the cells, and the small differences between these isoforms made it hard to design specific qPCR assays or really to deduce much about the expression using RNA-seq data. So we borrowed a trick from the yeast genetics toolkit, and we used CRISPR-Cas9 to tag TIN2 in human cell lines. So we just cut right by the ATG start codon and added a MIC tag at the beginning of the gene. And when we did a Western blot on some of these positive CRISPR-Cas9 clones, we were really surprised to notice 3 bands instead of the expected 2 for the 2 TIN2 isoforms, which just didn't make a whole lot of sense to us. So we cloned the short and long isoforms and tagged them, and also cloned the full-length gene and tagged that. And so we could see that the unknown band was falling between TIN2 short and TIN2 long. And based on its size and some other things we know about TIN2, we thought it might be yet another isoform, so creating more questions rather than answering our isoform question. And we couldn't really predict what that might be, so we did a targeted Iso-Seq of the TIN2 transcripts in different cell lines. So we just reverse transcribed TIN2 with this adapter sequence and did a PCR amplification with a primer in the TIN2 5' UTR and then to the adapter sequence, and submitted them to the core at Johns Hopkins to sequence with PacBio and then analyze and align these transcripts. And so we did this in 5 different human cell lines and 2 different mouse strains, which I should have mentioned before only express 1 TIN2 isoform, and that's exactly what we saw. So here's just an example from one of our cell lines. We got a lot of reads that map back to the TIN2 gene. So I'm showing them, each line is a transcript, and navy is a line sequence, and gray is what's skipped. And we saw a lot of rare but recurrent exon skipping events and some random intron retentions, and some things that look like they might be alternative polyadenylations. We're not sure if that's real or not or what that means, but we also saw 3 predominant isoforms when we analyzed the data. So we saw TIN2 short, we saw TIN2 long, and then we saw this new isoform, which we're calling TIN2 medium because we're really creative. And that is basically the same as TIN2 long, except that it retains the last intron, which has a little bit of coding sequence before it hits a stop codon. And so what's interesting about TIN2 medium and TIN2 long is they both retain that conserved patch of amino acids that TIN2 short doesn't have. So we cloned these cDNAs and expressed them in HeLa cells, and we saw that they all do localize to telomeres. So you can see the TIN2 isoforms in green overlap with TRF2 in red, which marks the telomeres. And through some assays I'm not going to get into today, we saw that they seem to be functional and protect and function at telomeres in vivo. So back to this question of, does TIN2 work with this telomerase processivity factor in any sort of way? So I'm not going to get into this assay too because it's a little hairy, but if you want to talk to me about processivity, I'd love to. So you can see in this graph that in blue, TPB1 and POT1 with telomerase is a lot more processive than TPB1 and POT1 without telomerase in orange, or with the processivity mutant in red. And when we added TIN2 to these assays instead of GFP, each of the isoforms was able to further stimulate telomerase processivity above that provided by the known processivity factor. And this is dependent on the presence of the TPB1 POT1 factor. As you can see, there's no change in these red and orange bars. So we concluded that TIN2 is functioning with TPB1 and POT1 to stimulate telomerase processivity and might actually be completing the processivity factor. And this would be a candidate for further study to see if the different isoforms might have different effects on stimulating telomerase in vivo. So sort of to summarize this, we found this new isoform of TIN2 using a combination of CRISPR and targeted Iso-Seq, and this had been missed in previous studies, but all 3 TIN2 isoforms that we know about localize to telomeres and can stimulate telomerase processivity, and their only difference is this variable C-terminal extension down here. So we think that it might actually hold keys to TIN2 function in vivo, maybe through a novel binding partner or post-translational modifications, and that is an avenue for further study. Might be interesting to know also if they have different expression in different tissues. And I might have gone through that a little quickly, but I'd like to acknowledge everyone at Hopkins that helped with this study, as well as my current PI at MIT who let me come here. Yeah, that's all.